Okay, I'd like to thank the organizers for giving me the opportunity to speak here today. So, basically, I lead a company called Nimura Pharma. We're based in the UK, and we develop drug delivery and diagnostic platform technologies. There are two parts to the business. One is drug delivery, which is all to do with putting drugs into the skin. And the other aspect is about diagnostics, which is taking analyze out of the skin in order to measure it. The diagnostic arm is one which is due to be listed on the NASDAQ later on this year. So what I want to do is give you a brief overview on the methodology that we adopt to develop the technologies that we have. So initially we look at unmet clinical needs. And we then evaluate the activity in the field, which means looking at what a lot of the multinational companies are developing and also where investors are putting money in terms of new technologies. We then identify the gaps and we go out there to develop intellectual property to meet the unmet needs and to fill those gaps. And we then go out to validate that platform technology that we've developed and develop a proof of concept, and after that we develop a clinical program and the commercial manufacture. So what I wanted to do today was really give you three distinct methodologies, three distinct technologies that we've developed using this approach. And hopefully you will be able to take from that the different approaches that can be adopted in order to develop a new platform technology and therefore a new product. We have a number of products currently in development. Some of them relate to transdermal patches. Now, most of you may be familiar with patches like the nicotine patch. So if you look at the first 4 products or 5 products there, these are essentially very similar to the nicotine patch. But in this case, they have drugs for various conditions like Alzheimer's, Parkinson's, pain management, etc. And then we have the diagnostic platform technology, which I will come on to later. So the first example I want to talk about is a product that is already on the market. So you might ask, well, where does the innovation come into on this? Well, what we identified was that this is quite a significant market. Currently, there are more than the sales of a particular patch product for Alzheimer's is in excess of $1.5 billion US, and that's projected to rise to possibly double that within the next 3 or 4 years. So what we identified here was that there was a gap in the market in that if you look at the average generic drug, there are probably 20, 30, 40 versions of that drug on the market. In the case of one particular drug, which is for diabetes management, there are in fact more than 100 generic versions. However, here, given the specialized nature of transdermal systems and the intellectual property constraints, what we discovered was that there were probably going to be no more than 3 or 4 players on the market for this particular product, although it may be a generic copy. So we went about to reformulate the product. And once we had completed the proof of concept, we then licensed out that patch technology to a multinational generic company. So although it's an existing drug, by virtue of innovating with respect to the formulation, we were able to develop a product which we could then license to a multinational, which is now being entirely funded by the licensee and which will then reach the market in a couple of years from now. So that's to give you one example of how innovation can be, if you like, implemented even for products that are already on the market, just by virtue of reformulation, as is in this case. The second example I wanted to touch upon is microneedle technology. So microneedles are, if you think about needles and syringes, you have one big needle at the end of a syringe. So what microneedles are is basically either dozens or hundreds of needles on a patch. And they're so small that you virtually cannot see them. And they range in height from anywhere between 0.2 millimeters to a millimeter and are generally pain-free. The biggest advantage of microneedle technologies is that although with patch technologies, we have a number of products on the market. Over 20 years of research has led to only about 14 molecules being currently on the market in patch form. But there are probably over 50, 60, maybe even 100, some say, drugs that would benefit from transdermal delivery. And if I give you two key reasons why that is, one is to do with compliance. So if you take the case of the Alzheimer's, for example, if you imagine somebody who already has memory impairment and they have to remember to take a medication 2, 3, 4 times a day, it doesn't bode well. And therefore, if you have a patch that can be applied once a day or maybe once a week, you can automatically see from a patient compliance perspective, it has significant clinical advantages. But in addition to that, there are significant benefits to delivering drugs through the skin as opposed to, say, orally, from the point of view of reducing side effects, and that's to do with the way the drug is metabolized. So microneedles provide a means of delivering a very wide range of drugs through the skin. In fact, you can deliver virtually anything through the skin using microneedles, because at the end of the day, you are breaching the barrier of the skin and you are putting the drug directly into the skin. However, what we discovered was that even despite its advantages, there are some significant limitations. So this is just a quick slide in terms of the way microneedles are used. Very quickly, you can coat the needles with drug and apply them to the skin. Or in the other extreme, you can actually have microneedles that are hollow and you literally infuse the drug through the skin, through the bore of the needle. So again, instead of the one needle, you would have an array of needles. However, if you think about it, you've now got an array of hundreds of needles. So when you apply that to the skin, I think most of you may be familiar with the bed of nail effect, whereby because of the force distribution over the surface area, the needles no longer penetrate the skin. So from a regulatory perspective, one of your fundamental hurdles is how do you guarantee that each and every needle has penetrated the skin to the same depth for the duration that is required for the drug to be absorbed into the skin? And this is a fundamental problem, despite everything that is going on in the clinics today, where in controlled clinical conditions, people are able to demonstrate that you get drug delivery and it's efficacious, etc., etc. Once these systems go out into the market, you've got the mass population who have to use this, and these are unskilled, untrained people, and there is a lot of scope for error. So the question is, how do you ensure that if you have a number of needles, each and every needle goes into the skin to the same amount, reproducibly, irrespective of who administers it? But another key problem with these systems is that a lot of the microneedles are produced from the drug itself. So if the tip of the needle is, say, compromised, then how do you guarantee that that needle is actually going to penetrate the skin? So these are quite fundamental issues, and there were a number of companies already in the market before we started developing technologies. And what we set out to do was we said to ourselves, well, okay, these types of systems will potentially replace a lot of the conventional methods of drug delivery in the future. But we gave ourselves a set of criteria that we wanted to achieve. First of all, we wanted to ensure that irrespective of who administers the system, it should be independent of that person. The delivery should be instant. So you shouldn't have to apply a patch and wait for 10 minutes or 1 hour or 24 hours for the drug to be delivered. We wanted that delivery to be instantaneous. So you apply it, take it away, and it's done. And if you say you're going to deliver a certain amount of drug, that is the drug that should be delivered. So we set about with those criteria in mind, and we then developed another platform technology, which we call the solid dose injector, which is currently being used for various molecules are being evaluated with it. And essentially what we have here is a hollow micropellet which is applied to a microneedle. And the needle is responsible for piercing the outer membrane of the skin, and then the drug is delivered instantaneously with what is a disposable device, which is quite cost effective as well and operator independent. And so here you have a platform for delivering quite a wide range of molecules. So I talked earlier about determining clinical need or determining an unmet need. So here in the area of vaccines, we established that there are a large number of products that are highly unstable in the liquid form. So if we can convert vaccines into a solid dose, then that cold storage and management chain is no longer an issue. Secondly, self-delivery suddenly becomes possible, but also with vaccines, because of the higher density of immune-stimulating cells in the skin, you can actually achieve a far greater response to a smaller dose, to the extent that with some vaccines it's been shown that with 5% of the dose, you can actually get the full efficacy. And in terms of validation, we have a British government-funded project whereby we're looking at taking a triple vaccine, converting it into a solid dose, for the reason that I've mentioned. So, finally, I want to talk about one last platform technology which relates to our diagnostic platform. And this is to do with diabetes. Currently, there are in excess of 350 million diabetics in the world. In Saudi Arabia alone, there are over 2.5 million diabetics, and that figure is due to rise, or set to rise, to over 5 million by 2030. The prognosis of diabetes is pretty poor. It ranges from people going into hospital, coming out without their limbs because they have to have amputations, blindness, kidney failure, cardiac issues, and various other complications that arise from excursions in your blood glucose level. So the question is, if you were able to monitor your blood glucose levels on a more regular basis than what is done by finger pricking, are there any benefits? Well, there are a number of studies that have been conducted which show that if you are able to measure your blood glucose levels on a more frequent basis, and you're able to maintain the control over the excursions, then indeed there are very significant benefits, to the extent that if you reduce your HbA1c value by about 1%, you almost halve the potential for some of the complications that arise. So the technology that we developed is essentially taking a patch technology, which instead of now putting drugs into the skin, non-invasively, so without needles, without injections, without taking blood, it extracts analytes out of the skin. It then measures it on board the patch, which, as you can see in the diagram there, in the picture there, the device at the moment is like a watch, and the patch is underneath it, and is connected to the device. Clinically speaking, even if you were able to get one reading per hour, that is still significant and that is still beneficial in terms of a person being able to determine what lifestyle activities and other parameters, such as food intake and exercise and things of that nature, how it affects their blood sugar level. And so the idea is that with this particular system, we take measurements 4 times an hour. We can do it less, we can do it slightly more, but that's deemed about to be the optimal. And right now, in terms of validating the technology, again, going back to my original hypothesis in terms of the procedure that we follow, the very first validation that we did was looking at government grants. And again, in this case, we received 3 or maybe 4 actually competitive government grants that total in excess of $2.5 million. And that's a strong validation, because what you have there is an independent body who have absolutely no interest in your technology, and they basically have a scientific team, a technical team, an intellectual property team, a team who looks at commercialization, and they have to tick all the boxes. But not only that, you are competing with a number of other players on the market. So not only do you have to tick all the boxes, but you also have to compete with your peers in the industry. And so it's a very strong endorsement to the potential value of your technology. And that's how we generally go about validating early stage our technology. And beyond that, of course, once we have secured the intellectual property, we're looking for licensing deals. And in this case, we already have a license for the UK and various other markets that we're looking at. But each market has its own intricacy in terms of the way a product like this would be marketed, and therefore we tend to look for multiple partners rather than a single partner in any one jurisdiction. So hopefully what I've done is given you an idea of 3 different methodologies, 3 different approaches that we've taken to developing technologies, right from completely new innovation that we've developed, starting from products that are already in the market. Okay, well, thank you very much for listening.